It's my pleasure to introduce today's Wednesday afternoon lecture speaker, Dr. Julie Segre. And this is also the annual G. Burroughs Mitre Lecture. Just to remind you, if you don't know who that was, he was, in fact, the first director of the NIH laboratories and clinics from 1960 to 68. And so this particular lecture put in place to recognize those contributions. Julie is currently a member of our intramural program, and we're very proud of that. Dr. Segre got her undergraduate degree in mathematics at Amherst, her PhD in biology at MIT. Her mentor was Eric Lander, and I have been told she was the very first graduate student, so that got things off to a pretty good go, I would imagine, considering the numbers that have passed through the Lander lab since then. She then went on to do a postdoc at the University of Chicago in the very distinguished laboratory of Elaine Fuchs. So it'd be hard to beat that for a pedigree. But of course, it's what did she do then? Well, what she did then was to come here to the National Human Genome Research Institute, where she is now chief of the Translational and Functional Genomics branch, and has done spectacular work, beginning with biology of the skin, and then the microbiome emerged. And I think it's fair to say that she has become the lead investigator in understanding the role of the microbiome in both normal and abnormal circumstances involving human skin. The GI tract gets all the attention for the microbiome, but it's involved in lots of other places, and certainly her work has demonstrated how important that can be for conditions like eczema. She has also immersed herself in this particularly unique community that also contains a research hospital, the Clinical Center. And working with her buddy Tara Palmore has taken this investigation of outbreaks of organisms, particularly those that are resistant to all known antibiotics, to an entirely new level of sleuthing to figure out sources and transmission. And that has led to numerous publications that I think in the field of public health are seen as absolute landmarks. It has also led to recognition in the form of an NIH Director's Award for tracking the Klebsiella pneumoniae outbreak, and maybe most notably, that she and Tara were recognized by the SAMMYs, the Service to America Award, as the Federal Employees of the Year for the entire federal government back in 2013, which, as you can imagine, makes us all very proud. We're also delighted that she is joined today by her daughters, Anna and Emma. Thank you for coming and finding Missouri Auditorium. And I imagine this will be an exceptionally special kind of afternoon for all kinds of reasons, but your presence would be certainly one of them. It's been my great pleasure to have a chance to work with Julie on a wide variety of fronts. And before I ask her to come and join, I want to say one other thing, that she has been incredibly helpful and significant as a wise advisor in the whole area that we are all now wrestling with, namely the problem of sexual harassment in the workplace. Julie, in her roles as member of the NIH Equity Committee, as part of the ACD Working Group on Anti-Harassment, and many other ways as an advisor, has helped, I think, all of us, particularly the men, to understand just how important it is that we face this issue in all of its manifestations, and recognize the need for culture change if we are truly going to see our workplace become a place where everyone feels safe and is welcome. So, Julie, thank you for that. So without further ado, please join me in welcoming Dr. Julie Segre, who is going to talk to us about microbiome, friend and foe. Julie.
Thank you. Okay. Hi, it's really a great pleasure to be here this afternoon and to honor Dr. Mitre for his contribution. We all know that we got to just come to the NIH, and this place had been built by giants before us. And especially for someone like me, I got to just come here and really just get switched on and have opportunities that I wouldn't have had anywhere else to be involved in this type of clinical research. So I'm going to share with you today my journey to study the human microbiome and the linkage from working on the Human Genome Project as a graduate student, which I thought was going to be the most fun I ever had, but to recognize, so when I started in the graduate program and started with the Genome Project, which was considered unconventional at that time, I really thought that we would find the gene and then we would fix it with gene therapy or something. And what I've come to see in the last 20 years is really that there can be a lot of changes in human health, just even by how we manage the microbial communities that live in and on people. So cystic fibrosis would be one great example where we've made tremendous changes in the care of cystic fibrosis patients, although Dr. Collins was part of the original team to clone the CFTR gene. A lot of the progress has also come just in managing the microbes that colonize the lungs of the CF patient. So I'm going to talk today about what has ended up being, again, one of the most fun projects I've ever been part of, which is the Human Microbiome Project, which really got jump-started by the Common Fund, but has now been integrated across multiple institutions. So when I say the human microbiome, what do I mean? I mean all of the collection of microorganisms and their DNA that live in association with the human body. The fungi, the eukaryotes, so an example in the skin would be Malassezia furfur. The archaea, we have not found them on skin yet. Bacteria like Staph epidermidis and viruses like human papillomavirus. So the microbiome is going to include all of those microorganisms. And just to give you kind of a perspective here, this is the seminal work from Carl Woese that was done also and continued by Norm Pace. Here you're seeing the phylogenetic tree of life. And so we are here as animals. We're here as animals. And I'm going to talk, about the entire tree of life. And when I talk about bacteria, you probably will get it that when I talk about how there's a difference between a propionibacterium and a Staph epidermidis. And that would be like the difference between, a bunny rabbit and a human. But I'm even going to talk about how much difference there is between different strains of Staph epidermidis. And you have to keep in perspective that could be like, we all accept that there's differences between different humans. And we have that kind of granularity in the microbial world too. So if you have a Staph aureus infection, one of the first questions is it oxacillin resistant or methicillin resistant? And that's genetically encoded. So it's those kinds of differences that are going to exist and be distinguishing between different strains of bacteria. So really just continue to think about this kind of tree of life. And I'll just kind of start with, there's a lot of these comparisons being made. I'm just going to kind of round out and sort of talk about it in the different ways. So one of the things that people talk about is, that humans are superorganisms, that we have human cells and we have microbial cells, and we live together as communities, and we evolve together. And so some of these numbers have been calculated. So if there's 30 trillion cells, human cells, in a 150-pound individual, the calculation is that there's an equivalent number of microbial cells, bacteria. Actually, no one's calculated the number of fungi yet, but the equivalent number of bacterial cells may be slightly more bacterial cells. And the viruses, like including bacteriophage, would be even outnumbering those. So if you think about a 150-pound human, 2.5 pounds of that person are their microbes, like the bacteria that live in their guts. But this is really the number that's always kind of gotten me. So the human genome, and I think is part of how we are considering health and disease. So the human genome is 3,000 megabases. With 24,000 protein-coding genes. And I give you that there's all these, non-coding genes, and there's a lot of alternative splicing that can happen here. But let's just stick with this 24,000 number, and that each cell contains the same genetic material. Now, if you think about the microbes, so they have much smaller genomes. Like a bacterial genome is 3 megabases, which is going to have 2,400 protein-coding genes. But these bacteria have different genomes. And they can be quite different one from the other. So if you look at the entirety of the bacteria that live in a human gut, it's been calculated that that would be about 3 million genes. And I think also from an evolutionary perspective, we have an idea of how much you could change from one generation to the next with genetics of the human genome, and how much selection pressure could have an effect over generations. But the microbes are dealing on a different time scale. So when we start to think about these inflammatory diseases and people start to wonder, have we done something? And they always talk about it as the gene-environment interaction. And so then I've been thinking, where are the microbes in this gene-environment interaction? Are they part of our genetic material, or are they part of our environment? And they're right at that interface. So it could be, and those are sort of the theories that are coming up about why are we seeing this increased incidence in these autoinflammatory diseases, that it could be having to do with that microbial-human interaction, and that some of what we're doing between changes in lifestyle, antibiotic usage, could be altering the microbial communities, and that when we think about how much evolutionary pressure and selection could have been done, between, myself, my children, my mother, my grandmother. My mother's generation didn't really have access to antibiotics, but now we've been, having generations that have antibiotics given as even as small children. So my talk today is really going to focus on the human microbiome and what is its contribution to health and disease. And I'll just throw out there this sort of, NIH is all organized around these institutes. And is the microbiome maybe going to be the 12th organ system of the human body? Because it plays these crucial roles, tuning the immune system, aiding in digestion, and providing protection from pathogens. Examples would be sort of the association with inflammatory bowel disease, the changes with diet and obesity, or with pathogens like C. diff associated diarrhea and colitis. So my wheelhouse is this, genomics. So let's talk about how we're going to really characterize a microbiome. And, classically, the way we would characterize the microbiome is culturing. But I'm telling you that there's like 10 to the 14th cells. So how are we going to count those? And more than that, a lot of these microbes are fastidious. They're very hard to grow. They're not going to grow as single isolates. And we don't know how to cultivate them. Actually, that's one of the remarkable things about the sequencing these microbes, is that we can then learn what are their metabolic needs and actually develop better ways to culture them. But so this culturing, this is going to be really hard. So the sort of genomic wheelhouse, building on the work, as I was saying, of the original Carl Woese and Norm Pace studies, is to use signature genes from these bacteria and from these fungi. So we'll take a swab and we'll immediately make DNA from it, and then amplify the DNA in this method with primers for the signature gene, like the 16S. So we'll get a 16S from all the different members of the community, and then computationally, we can align those sequences and do a taxonomic classification. Because the finding from Carl Woese and Norm Pace is really that there's a similarity among bacteria that belong to the same phylum, the same order, the same genus, the same species. And you can use this to taxonomically classify them. So this would be a great tool, but I think one of the first things we always ask when we have these new genomic tools is this going to make a difference? So this was the initial experiment that I did with Pat Murray when he was director of Clinical Center, Laboratory Medicine, where we did a side-by-side comparison and swabbed skin, and half of it went to my lab directly for direct sequencing, and the other half, Pat, wearing his Clin Micro hat, tried to culture as many things as he possibly could from that site and then sequence them. And these are the kinds of results that we got. So if we swab the side of the nose, we, from culturing, are going to get a lot of Propionibacterium and a lot of staph. I mean, these are going to be like the lab weeds. And what we're missing that we can see here are these Corynebacterium and other Actinobacterium. And if we do the same comparison from the navel, we're seeing an even greater expansion. So in this case, Pat then tried to trick it and add some, tween to provide the food nutrients for the Corynebacterium, and they could grow a couple. It's not that we can't grow these, but we're basically outgrowing them with Staphylococcus and other firmicutes. So that really sort of launched us that we were going to get data that would be different from culturing and would be more quantitative and, in fact, would be less work. And with that, I launch into part one of my talk, which is to talk about our friends and the studies that we did on the healthy skin microbiome. And these studies were really made possible by the enormous intellectual firehouse of, the intellectual contributions of the dermatology department. And I couldn't have done this without John Vogel, Mark Udy, Maria Turner, very importantly. And all of these studies will now continue and were designed and have now been continued with Dr. Heidi Kong. So Heidi and Maria's question was just, let's think about the human body as an ecological system. Because as dermatologists, they have this pattern recognition where they're looking at rashes and they have this topographic landscape in their minds when they're thinking about disease. And the question is, does that correlate somehow with bacterial communities? So Maria and Heidi designed this where we're looking at body sites where we have a predilection for disease that is associated with microbial communities, and we ask, first we're going to ask, what is the bacterial community of those body sites? This was really the study that kind of really launched us. And we're looking here, and we've defined these body sites as oily, moist, and dry. And we see on the oily sites that there's this preponderance of the dark blue Propionibacterium. And the moist sites have more of the Proteobacteria, and the dry sites have this greater diversity. And this is really coming at this idea that humans are ecosystems, and that the bacteria are really utilizing the humans as their food supply. So the bacteria are going to live here because they can find the nutrients that they need. And what we saw was that the microbial communities were specific to the body sites. So the greatest similarity we see is amongst the oily sites. But really, so for example, the bend of my left elbow is most similar to the bend of my right elbow, because this is the same ecology. And after that, the bend of my left elbow is most similar to Dr. Gottesman's, because that's the body site, and that's the resources that would be available to those bacteria. And the bend of our elbows are going to have more similar communities than the bend of my elbow would be to my underarm, because this is a sweaty site, and this is a moist crease. And that really does help to even understand that why we might see microbial communities, similarities, and see the predilection for human disease in certain body sites. We did a similar type analysis of the fungal communities. Now, instead of using the 16S gene as the signature gene, for fungi, we're going to use the ITS1 gene, which is between different ribosomal RNAs. And we can see here less diversity. It's a lot of Malassezia. And the greatest diversity that we see is on the feet. Again, not entirely surprising if you think about where do we see fungal disease. Athlete's foot, toenail infections, that's a place. Actually, we also think it might have to do with temperature. So I want to talk about, how do we use this kind of data? So this is a project that we've been working on for the last year about Candida auris, which is an emerging fungal pathogen. So you've never heard of it? I had never heard of it. I now spend 25% of my time worried about Candida auris. In 2008, it was identified on 3 separate continents. And the CDC has looked back through all the collections since then. Those are really the first cases. In 2016, the third patient in the US was found here at the NIH Clinical Center. By now, in 2019, we're having outbreaks in Chicago and New York inside skilled nursing facilities. These are places that we really have a medical need. Why are we concerned? Candida auris has evolved resistance to all classes of antifungal drugs. That results in the difficulty in treating bloodstream infections that result from it. Its primary site of colonization is the skin, where it can live for months and years. And as a result of colonizing the skin, the patients shed Candida auris into the environment where it persists. So you can culture it from windowsills. Testing requires 7 days of culturing on selective media. And so when I was down in Atlanta visiting CDC, they said to me, like, we need to work out something faster than this. So we started working with them on how to develop a molecular assay. We got samples from the first 2 outbreaks, and these are 2 of the hospitals or skilled nursing facilities. Here I've switched colors on you. The taupe is the Malassezia. This was one of the first patients identified there. You can see these patients still have the sort of taupe Malassezia, which we see as part of the healthy microbiome. But we can see here, this red is the Candida auris, and it can overwhelm the skin microbial community, and the skin community can shift towards more of a Candida-dominated community. We're working hard now to do longitudinal studies in these skilled nursing facilities to understand what are the underlying risk factors for becoming colonized with Candida auris. We're wondering if there are fungi that provide colonization resistance, are there bacteria, or are there other methods that we could use in these skilled nursing facilities that it's been really important for us, and us I mean Xin Huang, working with Mihalis and Yasmin's labs, to think about setting up, Xin has set up an animal model for this, because it's really hard actually to run clinical trials in skilled nursing facilities. And so we really need to know, will this treatment be effective? Would this be effective? And we're also trying to identify what is the primary site of skin colonization. So we're getting hundreds of samples from these outbreaks because we're still afraid that we might not be surveilling the right body sites to catch everyone. Okay, so we're really trying to develop this skin microbiome translational pipeline and not just do sequencing, but generate hypotheses from that. And one of the shortcomings that we had was in trying to understand, when I talk about the strain-to-strain variation, is to generate the hypotheses when we're only at the genus species level. And so it became really important for us to push the sequencing and to move from working with amplicon data, which I was showing you, to really working with shotgun metagenomic sequencing so that we could get to the strain level and we could look at the kingdoms, as I've been talking about the bacteria and the fungal interactions. And it is here at this point that one of the tremendous benefits of being part of NHGRI is really the ability to innovate and move through different sequencing platforms. And I wanted to highlight that in terms of how this has pushed forward these projects in the skin microbiome. Because at this point, it requires the technical analytic skills of Sean and Clay to figure out how to make sequencing libraries from these very low biomass samples and to avoid contamination, and the power of the NIH Sequencing Center to keep working with us to develop the data sets. And so we've basically moved through, I think, almost every instrument, moving from Sanger sequencing, which was our original one, to now doing our sequencing on the Illumina NovaSeq and generating 100 million reads per sample. So our pipeline is to extract the DNA and just immediately do sequencing from that. So we're not going to look at the human reads. We're going to take the non-human reads, and we're going to map them onto reference genomes, bacteria, fungi, viruses. And as well, we've done a lot of culturing in order to identify other reference genomes to improve the mapping ability. So we recapitulate the bacterial and the fungal representations that I showed you. This data gives us the first look at what is the relative abundance of the kingdoms. So you can see here that the majority is the yellow bacteria, but they're also on each of these body sites is a representation of the viruses and the eukaryotes. This analysis really pointed out to us the variation between individuals. So when you think about clinical studies, we have to think about what's the variation between individuals, because if you want to think about patients versus a disease state, you have to think about that the difference between healthy and disease has to be greater than the inter-individual variation. And here's where we really started to see how much variation there was between individuals in the microbiome. So for example, this is just the side of your nose. 3 different volunteers. And our study design is we sample the person with shotgun metagenomic sequencing. Heidi will bring the subject back a year later and then a month later. And what we saw was this kind of variation where this person has a large amount of viruses, Propionibacterium phage. But that's their microbiome. It's not that they're just having a phage bloom. They will stay, and that will be their state for the next year and month. Now, this person's side of their nose, they don't have much phage. They have a lot of this taupe-colored, which is the fungi, and they're going to stay that way. And this third individual has a mixture of bacteria and fungi, bacteria and viruses, and they're going to stay that way. So what we saw here was that there's a lot of inter-individual variation, but there's a lot of stability to the individual. And we see this also at the strain level. So I've sort of gone from like the highest level to the lowest level here to show you what kind of stability we see on the individual. So this is, again, those same 3 time points, 3 individuals. And now I'm just showing you the Propionibacterium acnes, the population of P. acnes, which is one of the dominant skin microbes. And you're seeing here that this is the phylogenetic tree for P. acnes. So these different strains can be 20% different from each other. And our 3 healthy volunteers would have 3 different combinations of strains. And by having multiple strains, they optimize the entire pangenome. But what you can see here is that they will have 3 different strain collections, but they will keep these strains over the year and especially over the month. So this kind of long-term stability really powers us to be able to do studies of disease. So I'm going to talk next about atopic dermatitis, which is a classic skin disease, more commonly known as eczema. Heidi's been interested in eczema for a really long time, in that it's a common disease, but it's increasing in incidence where the disease criteria haven't changed. And we're also seeing that the patients with moderate to severe disease, over half of them will go on to develop asthma and hay fever. This is called the atopic march. And so there's really this idea that possibly if we intervened on the early childhood, when we talk about the microbial colonization of atopic dermatitis, could we have benefits not just for this disease, but for the other atopies? And so that's a really interesting idea about microbial communities, is like when they travel together. Okay, so the clinical study is designed by Heidi, and she recruited 11 patients with atopic dermatitis and 7 healthy children, and did clinical assessment and collected clinical samples from body sites that are sites of disease predilection and other control sites. This would be an example of going from baseline. You'll see that the 2 most common sites are the bend of the elbow and behind the knee, 2 sites that actually do have very similar bacterial communities. And the assessment will be done with, Heidi will do it with SCORAD, the score of AD severity. And you can see that at baseline, their skin is not 0, but there's definitely these flares or episodic exacerbations, a dramatic increase in the AD severity which after treatment goes back down. So what we see in the skin microbiome of these patients is that at baseline, they're going to look a lot like the healthy control kids with a lot of diversity. But during the flares, there's a decrease in diversity, and that's driven by this increase in Staphylococcus. For some kids, it's an increase in Staph aureus, and for other kids, it's an increase in the Staph epidermidis. So we at the same time were culturing isolates from all of these patients, and when we colonized the kids, each kid has a unique strain of Staph aureus. Just one strain of Staph aureus colonizes each kid. It's different than the multiple strains of the commensal Staph epi. So each kid will be colonized with a different strain. And you can see they're quite phylogenetically distinct. Now we're going to test to see whether these strains are in any way driving disease, because we want to move beyond just associating to seeing if they have any effect in driving disease. And so we're going to use the strains from the atopic kids. Okay. And what we see is that when using an animal model that was developed by Yasmin Belkade, we're going to colonize the mice who are wild-type mice with an intact immune system and an intact skin barrier, and then look, and what we can find is that after just 2 weeks of being colonized with these strains of Staph aureus, we see that the animal model recapitulates some of the features of atopic dermatitis, including this epidermal thickening and the infiltration of immune cells. So these are the strains of Staph aureus that we're getting from the kids with atopic dermatitis, which all will result in this kind of epidermal thickening. And really, I think what's important here is that if I had just ordered USAA 300, which is the classic MRSA strain, order that from ATCC, that's dead in our assays. And so it's really specific to these strains that come from the patients, the direct isolation that's led to us being able to generate and then test hypotheses like this. So this is, sort of the model that we're trying to develop is this, sequencing and culturing to test, to generate animal models and test hypotheses. And it's not just us now. There's other people. The cohort of colleagues working on the skin microbiome, people are sequencing them and finding isolates and what's in the commensal strains, and testing to see if you colonize mice with one strain of Staph aureus, could you prevent other strains of Staph aureus from colonizing, much like how we're seeing the sort of revolution in thinking about peanut allergy and LEAP. And so I haven't talked about, and I'll kind of just move quickly through, we've talked about bacteria, we've talked about fungi. When we do amplicon sequencing, we are blind to the viruses because there's no signature gene for viruses. The only way to understand the viral communities is shotgun metagenomic sequencing. And even there, we don't have very many reference genomes, so we're still challenged. But here at the NIH Clinical Center, again, there's this astute observation with the primary immune deficient patients that a subset of them who have mutations in the dedicator of cytokinesis 8 gene had a lot of cutaneous viral involvement. So you're seeing HSV, warts, which are commonly associated with HPV, and molluscum. And so Heidi recruited these patients onto our clinical protocol. And mostly what I've been showing you is healthy volunteers who would have a preponderance, in the atopic dermatitis kids, who have this preponderance of the green bacteria here. When we started sequencing the DOC8 patients, these are 27 DOC8 patients, their skin is just covered in viruses. In sites where you see warts, but even in sites where you don't see warts. And we're finding a lot of papillomaviruses, also molluscum and polyoma viruses. And our strategy here, as you've seen before, is to take these shotgun metagenomic reads. Now we're going to subtract out the human, the bacterial, and the fungal reads, and just be left with the viral reads, and also what we call the dark matter, which are the reads that don't map to anything because they don't have a reference genome. And we now do de novo contig assembly and can assemble from that 8 kb contigs for the pox virus, larger contigs, which, when you blast them, are going to be known viruses and novel viruses. And I'll just tell you as one story that we really dug into the human papillomaviruses and found 205 new HPV types on these 27 DOC8 patients. We found 1 new HPV species, and we identified 25 types from that species. So I think with viruses, there still is a lot to be discovered. When we looked at our DOC8 patients, all this light blue, so this is like the bar charts now stuck on their sides. So I'm showing you the relative abundance. And this light blue, these are all reads that map to what are novel gamma HPVs that we identified in these patients. So if I might have told you before doing the study that there were about 75 HPVs on a child or on an adult that was a DOC8 deficient patient, now you have to like double and triple that. And with these new HPVs that we found, we're finding more on average of like 200 HPVs. So in conclusion for part 1 about our friends, our skin microbiome is part of the healthy all of us. And so the bacteria, I sort of tried to touch on and talk to you about how strains really matter functionally and how we have to understand everything from all the way from the kingdom approach all the way down to the strains. For fungi, work again sort of trying to understand the different kingdoms and how they interact. And for fungi, thinking about fungal pathogens, which I hadn't really spent much time before on. And viruses, lots more to be discovered. And in particular, really, I think that this field is going to move forward with these translational microbiome systems. And that's where I think we really have this amazing opportunity here at the NIH Intramural program between the clinical findings, developing the ex vivo, in vitro animal models, to really generate and test hypotheses. And I think that's, and to test those hypotheses generated from the human data and then use that to develop new therapeutic modalities. It would be really awesome if we would do first-to-man trials here on new therapeutic modalities for microbiome focused. So now I'm going to spend a little time and talk about our foes. And this was, again, really like working with Pat Murray on the skin microbiome. That's one of those great things about coming to the NIH. You think when you meet people and you really like working with them, you think, is there anything else we could work on together? And they always have something in their back pocket. Oh, well, I've been wondering about this. And so Pat started talking to me about hospital healthcare-associated infections and his experience with Acinetobacter baumannii, which I'm not going to talk about, and sort of wondering if there had been multiple introductions or if there were transmissions. And I just got totally gobstopped when I started learning more about healthcare-associated infections in terms of complicating 5% of hospital admissions, 2 million people affected annually, and really, one of the top 10 causes of death in the US. The economic toll, which I think Jim O'Neill from the UK has really done an amazing job working on that. And a lot of work has gone into thinking about the economic toll. And the societal toll about the public's concern about seeking hospital care because of hospital-associated infections. And I think it's really this personal connection that drew me in here about having people who are worried about that possibility. So we all kind of know this shoe leather epidemiology and John Snow, and what did he do? He broke the handle off the pipe. Well, how are we going to do that? And I think that we're going to do that integrated with genomics. I'm going to tell you briefly the story that we worked on and kind of how we've been tracking hospital infections. And this was the first case that I worked on with Dr. Tara Palmore and David Henderson. And there was an index patient who came into the hospital and actually was colonized with a Carbapenem-Resistant Klebsiella pneumonia, was put into the ICU on 2 different occasions. Then a month goes by, and then there's clinical cultures and surveillance cultures. And the question is, I don't think this is working anymore. Okay, oh, sorry. And what is Carbapenem-Resistant Klebsiella pneumonia? Why do people worry? Well, this does also make it to the list of urgent threats developed by both NIAID and CDC because of the lack of antibiotics that are available to effectively treat infections with Carbapenem-resistant Enterobacteriaceae. So the question was, as we have these first patients, is there a match? So was there hospital transmission, or is there no match? And that kind of decision matrix leads you into 2 very different directions in terms of how to respond. And at that point, the diagnostics were still kind of looking at these pulsed field gels, which maybe Francis and I are the only 2 people who've ever run pulsed field gels. But so this gives you this resolution where you know that these isolates are all sequenced type 258, but 70% of the healthcare associated Klebsiella pneumonia are sequenced type 258. So this does not give you the resolution that you need. So we sequenced the genomes of these bacteria, and we're seeing, we're looking now for single nucleotide variants, single base pairs that have changed. They don't even have to change the function. They just have to give us a signature so that we know that this is, this bacteria, and it becomes a unique signature for that strain. So there were multiple isolates from the urinary tract, and they all were identical. There was an isolate from the throat, where there was 3 base pair changes compared to the urinary tract. There were also isolates from the inguinal crease and from bronchial alveolar lavage, and those had 3 different SNPs. So what we assume from this is that these SNPs have evolved in this patient who has been colonized at this point for about 6 months, and that you have now this physical separation, and these strains have evolved independently. So that's important that we think these SNPs actually evolved in this patient and therefore are a marker of that patient. Because when we sequenced patients 2, 3, 4, 5, what we saw immediately was that patient 3 has the exact same genome as the throat isolate from patient number 1. And that patient number 2 has 1 additional change. In that meeting, which I think everyone still remembers, we're sitting there, we've gotten the sequence back maybe 4 hours ago, and we're saying, Tara, what we think happened is, patient 1 transmitted to 3, transmitted to 2, and she says, yes, that's exactly what I think happened too. Because patient 1 and 3, this is trace data of where the patient was every day in the hospital. Patient 1 and 3 overlapped in the ICU, and then patient 3 and 2 overlapped in the ICU. And suddenly we have genomic and epidemiologic data, which both would point in the same direction, but together, when you have independent confirmation, you have greater confidence in it. As the patients were identified, we continued to sequence every isolate, and that helped to identify where there were transmissions, but also where we could rule out transmissions, so that if patient 9 and patient 10 were identified 1 week apart from each other, and they had the same wound care cart was being used to care for them. But if we can rule out that they don't have the same isolate, then that's as important as ruling in transmissions. So again, the epidemiologic data would be based on where the patient was every day in the hospital, and that creates this sort of plate of spaghetti. Our genomic data where we continue to sequence every isolate, you can see that these kinds of SNPs that evolve and then are passed on, you can begin to see how that would create a genetic linkage. These SNPs would create a linkage block, which then becomes this linkage block and this linkage block and this linkage block. And while neither method alone is completely able to identify what were the transmission links, it's really this partnership and working together with the epidemiologic and the genomic data where we can reconstruct the transmission and use this information to understand what are the changes that we need. This is a more recent case that Tara and I were working on together with Anna Lau. So this is an unusual case where there are 6 patients who develop Sphingomonas infections, and I will leave this to the clinicians to talk about, but this is not something that any hospital, the NIH Clinical Center will investigate this kind of thing and get to the bottom of it. And so Tara's question to me was the same thing of, is this do we have transmission, or is there, an outbreak, a cluster of infections, or is this a polyclonal, thing where it's all different strains? So we get the 6 isolates, we sequence them, we assemble them, and then we say, how similar are these genomes? And this was a pretty quick turnaround and a pretty quick phone call to Tara, because 4 of the isolates are basically clonal. And over their 3 megabase genomes are 99.992% identical. And 2 other isolates are from completely different Sphingomonas species, Sphingomonas posimobilis, Sphingomonas yankoni. But the 4 of them, there is a similarity, and that's where we start to investigate. And in particular, this gold mine of the NIH Clinical Center, where we've stored the NIH Clinical Department of Laboratory Medicine has stored every isolate. So sorry, I forgot this. Let me just explain to you what Sphingomonas is. I don't have one of those pictures from the CDC and the NIH showing you how dangerous this is, because this is not typically considered that dangerous. It's actually it's considered low pathogenicity, but there are some features that are quite concerning about something like Sphingomonas. So they can survive in nutrient-poor conditions, so they can survive in water. And what that means is that they are not going to evolve antimicrobial resistance in that they are more typically going to pump out everything and then only selectively bring in what they need. So these bacteria, the 4 patients, those isolates were highly antibiotic resistant. Because the organism is just intrinsically antibiotic resistant. So Anna goes back in through all the records and identifies every Sphingomonas isolate that's been cultured from a patient going back over 10 years and pulls them all out of the freezer and tests them then with MALDI and with 16S and gives us the rest of the isolates to sequence. And we sequence these isolates, and we can find that going back till 2006, we could find our first clinical isolate with Sphingomonas carensis, and these are all still 99.992% identical to the isolates that are colonizing and infecting patients 10 years later. And so what we think is that actually this is resident in the hospital, or was resident in the hospital, because then at that point they started taking apart the different plumbing systems to find it in the sink, the water, and the pipes to then find it and to remediate the situation by increasing the water temperature, increasing the chlorine levels, and by actually physically cleaning those pipes. So those are the kinds of outbreaks that we can work on with combination of genomics and epidemiology. But there was another question that had been really concerning through all of this, and in particular with the Carbapenem-Resistant Klebsiella pneumonia outbreak, which was that a lot of these antibiotic resistance genes are found on plasmids. And there was always this question about if you have these high-level antibiotic-resistant plasmids, are they going to transfer that gene to an antibiotic-sensitive strain? And so that would be kind of when we talk about the nightmare, scenario. It's that the commensal microbes in your gut suddenly, even they become antibiotic resistant. So are we going to have like urinary tract infections that are untreatable? And that's always been the sort of where are we on the landscape here? But it was really hard to figure out because what we had at that point were all these short reads, and we couldn't assemble the plasmids. And it was at that point, then again, we had this genomic innovation, and NIST came back from our NIH Sequencing Center, came back and said, take a look at this. These are long reads, and we did our first genomes. The whole thing just comes out. It's like it just comes out as one piece. We were able to assemble immediately the plasmids and ask questions that had before baffled us. So patient number 5 in the outbreak was colonized with the Klebsiella pneumonia from that was the NIH Clinical Center strain, which contains this plasmid PKPQIL with the KPC or the Kleb-pneumo-carbapenemase gene. But that patient was also colonized with an Enterobacter cloacae that was KPC positive. And we wondered whether this was transmission of the plasmid from one strain to another. But when we got back those PacBio genomes, it was immediately obvious that no, it's 2 separate plasmids. This is an INC-N plasmid, and there was no horizontal gene transfer. This is actually a patient being colonized with 2 totally separate isolates. But that's not always the case. This is a really, NIH has, anything you want. I mean, the patients here are just, amazingly complicated. This is one patient that we've been working up for about 2 years now, where there was, on the first visit, there was a Klebsiella pneumonia detected that was carbapenemase resistant and had plasmids with the KPC gene. There actually were 2 different plasmids encoding the KPC gene. On the third visit, the patient was colonized with a Citrobacter freundii, in which we found the same plasmid as the Kleb-pneumo. And in fact, actually going back to visit 1, as I told you, they store everything here. We could find the Citrobacter freundii without that KPC plasmid in it. Another plasmid. At that same visit, we found a different strain of Kleb-pneumo that was carrying that same KPC plasmid and another KPC plasmid. We found a Klebsiella erogenus that had, again, the red plasmid. By the 4th visit, we had an Enterobacter cloacae with that visit, with that plasmid. And by the 7th and now final visit, we have a Klebsiella pneumonia, but the same strain, but now it also has this green plasmid that we had previously only seen on visit 3. So these would be plasmids that I would worry about, and we have the same thing with an INC-N plasmid that we've seen it in multiple species. So these would be the kinds of plasmids that you do have to worry about going between different species. So in conclusion, we really have just focused on how DNA sequencing is a powerful new modern microscope to identify microbial pathogens, and our hope that genomic sequencing of these contemporaneous and the historical isolates, which is really one of the hallmark features of the Clinical Center, we've used this to establish a national pathogen reference database together with colleagues at FDA and CDC of both plasmids and genomes. and that the NIH Clinical Center, these genomic epidemiologic investigations to distinguish multiple introductions, subclinical colonization, blooming with antibiotics, transmission networks. We've used all this to serve public health and to create a leadership role for NIH in really how to cope with hospital-acquired infections. My overall conclusions are really that genomic sequencing, in my mind, makes the invisible world of microbes visible and identifiable. And I hope that the understanding the microbiome will revolutionize what it means to be human. I've seen a lot of citizen science come out of this project, and what it means to be healthy, and provide new insights into how we're going to fight these microbial foes. And we're going to do that by cultivating our microbial friends, as well as by developing new antimicrobial strategies. Wanted to acknowledge, finish by acknowledging the enormous colleagues that I've had here at the NIH Clinical Center. And in particular, all of Project One has been developed with Dr. Heidi Kong, who's designed all the clinical studies, worked with me on all of the analyses. Dr. Tara Palmore, who led all the studies for as hospital epidemiologist, and Dr. Yasmin Belkade, with whom we've developed some of our ideas that I didn't present about the immunologic consequences of the microbiome. And shout out today, she was awarded the FNIH Lurie Prize, so congratulations. And to say that without these three, science might have gotten done, but it wouldn't have been done as well, and it wouldn't have been as fun. And they were always my first phone call. I've had fabulous collaborators at multiple institutes of NIH. And if you don't see your name up here yet, just come and talk to me, because we're always looking to work with new people and hope to see your name up there next time. But really, the acknowledgments go to my lab. This was the group that got us started on the Human Microbiome Project, and this is the group now. And of course, the one similarity between the two photos are Clay and Sean. And as well, just when we talk about Burroughs Miter, who built the NIH Clinical Center as the director, these have been the people who have really built NHGRI for me while I've been here. Thanks also Emma and Anna for coming today, and Nick. Oh, and now I'm happy to take questions, and thank you all for coming.
Yes, and if you're going to offer a question, please come to the microphone so people on video can hear the question. Start over here.
It came out of Sloan with respect to acute graft versus host disease in the microbiome. Do you have any comments on that? That seems to be the most profound use yet of the microbiome.
Yeah. So Eric Paymer and Schwauig Xavier set up a clinical trial for fecal microbial transplants, and that was funded by NIAID and their systems biology AMR initiative. And they are looking there to see if a patient, once they are reconstituted, so actually I think their primary outcome for that was C. diff colonization rather than graft versus host, because of the way they had to power the study. And they were seeing there that recolonizing the patient with their own stool actually was protective against Clostridium difficile colonization infection. And that's probably where we have seen the greatest benefit for the microbiome in general, is patients with recalcitrant C. diff to receive an FMT, a fecal transplant, from a healthy donor. That's really kind of been a lot of, also importantly, there have been no adverse events. So, imagine taking a drug that has no side effects and can cure you.
Okay, thank you.
Over here.
Yes. What does it mean to be human? And if you can't answer that, an easier question, when you see different plasmids in different species with an antibiotic resistance gene, one possibility is that nonetheless the gene has transferred from one plasmid to another by various mechanisms, and that must have happened at some point in history if they carry similar genes. So can you say maybe just by sequence differences in the antibiotic-resistance genes whether you can rule that out?
Right, so I'm not going to answer your question exactly the right way, but I know for myself, like when I'm trying to figure out how did Candida auris come to colonize human skin, I feel like I want to just take the genome and like shake it and say, I know the answer is in you somewhere. Like just tell me how did you figure out how to colonize human skin? And so, that is a lot of what we do. We have this data, and then we try to think, how did this happen? Like, phylogenetically, we can see when you look at a genome, you can find regions that have a different tetranucleotide frequency. You can see regions that are quite genomically different than the rest of the genome in terms of, these kind of genomic frequencies. And then you think, this must have come from somewhere else. So there are these little secrets that are left behind in a genome that we do try to use to figure out what is the evolutionary history of this organism. But in terms of what it is to be human, we are not human without these microbes. I mean, these are part of us.
You've done such excellent work here in terms of hospital epidemiology, tracking down the source of infection. One would hope that, since this has been published in prominent places, that lots of hospitals would be picking up those same skills in order to try to eliminate what is clearly a very serious problem of these outbreaks. How much of that has been happening across this country or across the world? Is it now in some ways expected that in such an outbreak there will be this sort of sleuthing using both genomics and traditional epidemiology to try to end the passage of what can otherwise be a life-threatening organism?
Yeah, I mean, this sequencing is now moving out into hospitals, and I think this is one of the ways in which doctors will begin to understand genomic information, as well as in the sort of ways in which we think about cancer diagnoses. And Dr. Palmore does receive phone calls, and people are very grateful for her advice on those kinds of questions. It is happening on a national level in the UK, in Israel, and there are now different mechanisms in place, the levers being pulled like, CMS and, in terms of when patients get readmitted and hospital, what we think of as hospital-acquired infections. So, yeah, I think that this is being taken up, and this type of genomic information is now being fairly widely utilized, although I would say still skilled nursing facilities, long-term acute care hospitals, those are places that I'm doing more and more work because I think those are some of our most vulnerable patients.
Over here.
On that topic, do you think that there's any objection from, say, hospital management to doing these kinds of studies? Because like in your example, technically somebody could say, well, this was a healthcare-associated infection, ergo the infection control procedures may be deficient. So there's maybe a conflict of interest there between providers and hospital management.
So we have these life-saving technologies where we're going to, reconstitute someone's immune system in order to cure them, and that puts them in a very vulnerable position. So I think there has to be some appreciation that this is a risk that hospitals are really working hard to medically manage. But you are right that this is something that the NIH Clinical Center has had real leadership on because they've allowed us to do, to publicize these types of investigations and to serve public health with that.
And many hospitals have been reluctant to be as obvious about what's going on for reasons you've just pointed out. Last question.
So, are you following up the evolution from fetal stage versus the adult stage?
We've been involved with a number of birth cohorts, and we've been particularly focused, and those are done in collaboration with other people because we don't recruit infants here. But I think, we've been very much looking at early time periods and the onset of atopic dermatitis. Could we even see a molecular microbial signature at, 1 month, 3 months? I think there is a potential for that, and there's a lot of people who are interested in that because of the idea of microbial transmission. And I think that these first 2 years of life are really somewhere where the microbes in the immune system are engaging with each other, are really critical time periods, but we don't have data on that now.
In patients with cesarean section, in the fetus or babies, there is a major difference, and in fact they use some of the lactobacillus for transfer. So you think this localization is happening depending upon the delivery system?
There are some associations. They're not totally clean. There are also babies born vaginally who have a microbial signature that looks like that. I think that's an area that will definitely get a lot of research in the next few years.
Since we are feeding them, are they helping us in burning calories?
Yes, I think, but we're also overwhelming the system at this point with our amounts of sugar that we can eat. So thank you all so much for coming.
Yeah.